biologist at the International Center for Coral Reef Research and Restoration on Summerland Key, which is a part of Moat Marine Lab, an independent, non-for-profit lab and aquarium. I'm a marine and evolutionary biologist, and I'm mostly interested in understanding how organisms cope with environmental change. And currently, I'm working on developing interventions based on sexual reproduction for generating more resilient coral offspring that can be used for coral reef restoration. So a little background, I'm sure this is not new for everyone here, but what is a coral? Of course, it's an animal, and it's a colonial animal. So it's made up of thousands of individual polyps. Each one of those polyps has a ring of tentacles that have stinging cells that are used to catch prey to bring to the mouth of the polyp, so the organism can actually feed itself. And all of the polyps are connected and share resources and nutrients. So if one polyp dies, not the entire animal dies. Some of the different types of corals that you might see in the Florida Reef Tract, we do have a diversity of species, not as diverse as, say, the Indo-Pacific, but we do have a variety of fast-growing branching corals. Sorry, I'm going to step down. Like the elkhorn and the staghorn, we also have a variety of brain corals. And within those species, you can have different colors, patterns, shapes, morphologies. So for example, these are all brain corals. This is one species, two different types of the same species. And the same thing here. We have flower corals, and we also have star corals. So what is a coral reef? Well, it's an underwater ecosystem created by these reef-building corals that secrete calcium carbonate skeletons that form the basis of the reef. And this reef creates an ecosystem for other reef-associated organisms. Let's step back a second, though. Why, in general, is the marine environment important? Well, it does cover 70% of the Earth, and therefore is a global life support system. It regulates the Earth's climate and atmosphere by absorbing heat and CO2 from the atmosphere and moving the heat around the globe using air and water currents. Why are coral reefs important? They constitute a mere fraction of the marine environment, but have a profound value and importance on a global scale. In essence, they provide a wide range of economic, environmental, and ecosystem services. They provide jobs. They're a source of tourism and recreation. They provide natural barriers and coastal protection to those living on the coast. They occupy less than 1% of the marine ocean surface, but they sustain more than 25% of all marine life. This makes them hubs, very important hubs of biodiversity. They provide food, habitat, shelter to other coral reef organisms. They're valued at $10 trillion on a global net worth. And most of the air that we breathe is produced from marine algae and marine microbes that come from the ocean and associated with coral reefs. They also provide food to humans. And therefore, coral reefs support 500 million people every year. They're also considered the medicine chests of the 21st century. So treatments for a number of these diseases listed up here have been sourced from organisms or coral reefs themselves. They also, people have also been able to develop novel building resources from organisms that come from coral reefs. But what are happening to our coral reefs? Well, to put it simply, they're dying as the environment rapidly degrades around them. To put it even more simply, the environment is changing faster than what corals can keep up with. Why is this happening? Because the Earth has absorbed the majority of the heat produced by human activity, mostly from greenhouse gas emissions, within the last 50 years. If the ocean did not do this, our average global temperature would be at 122 degrees. For us in Florida Keys, it's already damn hot all the time. Maybe it's not making a difference, but for the entire globe, that's pretty hot. So as a result of the ocean absorbing all of that heat and CO2, the ocean itself is changing and changing very rapidly. So what are some of these changes? You have warming ocean. The temperature of the sea surface is increasing, and this is causing a lot of the corals to become stressed out and starve. With melting of the polar ice caps, you have sea level rise. That can increase sedimentation and kill off corals. You also have changes in storm patterns. We're getting more frequent, more common, more intense natural disasters. And for corals like the branching corals, this is basically demolishing populations. They cannot withstand the intense physical forces associated with these natural disasters, and they're being decimated. Changes in precipitation. With agricultural runoff, we're getting increase in pollution and harmful algal blooms, and I'm sure everyone here has heard about red tide. We also have altered ocean currents. So the currents that normally would take larvae, coral larvae or food and move them around different regions, those are being disrupted as well. And then finally, with the absorption of carbon dioxide, you have ocean acidification. So organisms that secrete calcium carbonate shells or skeletons like corals, they either can't secrete them as quickly or they're dissolving. So as a result, we've had rapid and global loss of coral cover. 50% of the world's coral reefs are gone. On the Great Barrier Reef, it's also 50%. In the Caribbean, it's up to 80%. And on the Florida Reef Tract, we've gone from 98% in coral cover to less than 2% with the main reef-building species. 75% of the world's reefs that are remaining are threatened, and it's been predicted that 90% will be gone by the year 2050 if human intervention is not imposed upon now. To me, this is extremely alarming because coral reef ecosystems are some of the oldest living ecosystems on our planet. They've been around for hundreds of millions of years. And the fact that we've lost such a significant amount within the last 40 years, to me, is very alarming. So why is this such a problem for corals, though? Why are they dying so quickly? This is in part to the life history of corals themselves. They're long-lived, they're slow-growing, they have long generation times, meaning there's very few reproductive cycles, and therefore they cannot adapt as quickly to the changing environment as other organisms. Also, they're sessile. They can't move. They can't walk and fly, drive, run, whatever, to a habitat that has better conditions. They're stuck. So for example, I took a picture on your right, is a picture I took last summer. I was on a mission called the Ocean X mission, which was surveying the Florida Reef Tract. And I'm just going to show a few pictures of what a healthy coral reef would look like and what I'm seeing when I'm out in the ocean on the Florida Reef Tract doing my job. And these are not before and after, it's just a comparison of healthy versus what I'm seeing. So this picture I also took last summer, and what we're seeing as the corals and the coral reefs are dying, they're becoming covered in macroalgae, this really thick, slimy algae. And this prevents other organisms like corals themselves or other beneficial organisms to actually survive on the reef. Also, larvae, coral larvae swim, and at some point they find a nice place on the reef to settle down and attach. But with these stones and the reef structure covered in macroalgae, there's no place for them to settle down. In some cases, it's just flat and barren. And that's a problem because reefs provide rugosity. They provide three-dimensionality, which is shelter and habitat for other organisms. And without that three-dimensionality, you're not going to have shelter for fish or marine invertebrates. And in general, you can just see the reefs, they're not thriving. They're not colorful, they're not very dynamic. You see something kind of bright here, right there, you're like, oh, maybe that's something. Yep, it's disease. So the Florida Reef Tract is experiencing the deadliest disease outbreak ever recorded for any reef basin in history. Millions of corals have died within the last few years. It's called stony coral tissue loss disease. It has 100% mortality for some species. And these species, they grow very slowly, some of them millimeters a year. And it takes them centuries to reach the size of maybe this box here. And this disease is so virulent and deadly, it can cause a coral that took centuries to grow, it can die within a matter of weeks. The disease will just take over. And what you can see here is, this is here, healthy tissue, followed by the active disease margin. Basically, the tissue has just sloughed off, so all you see is bare, brand new, not brand new, but bare skeleton. And then behind it, other things start colonizing, so you can see a light green layer, that's algae, and then this died a while ago. It's now being fouled by other algae. So you see this everywhere we go. On the top, you can see non-disease colonies, and in the bottom, you can see corals with dark patches, that's living tissue surrounded by disease margins. This is sort of showing like a time lapse of a different species. These are 4 different corals, but they showed the different stages of the disease. So you have on the top left, top healthy, it's infected, then you have a small patch, and then that probably died within a few days and probably took several years to grow. And that's probably about this big. Corals can get reinfected. They can get multiple lesions. So you can see in the bottom left, there's a coral there with multiple active lesions. And then probably some of the saddest things that I've seen is Montastria cavernosa. These are the mountainous star corals. They can grow 15, 20 feet high. They can literally be underwater mountains. And they probably take 500,000 years, if not more, to grow to the size that they are. And in the right side picture, you can see the dark patch in the middle, bordered by white. That's active disease just eating across the colony. So what you see is on the top, a healthy followed by infected, and then a dead colony. And on the bottom, like I said, these are 15, 20 feet high. So they've been around for centuries and centuries and centuries. And if you look closely, you can see little white patches. Those are active lesions. Within a few short months, these colonies are probably going to be completely wiped out. So we're losing diversity, we're losing structure, we're losing a lot of services that these coral reefs provide. So what are we doing to help these corals? Mote Marine Lab is dedicated to resilience-based coral reef restoration. What does that mean? Well, first of all, we identify the most resilient strains to use for restoration. These can be strains that we identify that are fast growers, disease resilient, or stress tolerant. So the Florida Reef Tract has experienced a number of stressful acute events over the last years, cold water snaps, hot heat stress events that led to coral bleaching, disease, and we've lost a lot of corals. So a lot of what you actually see out there are the survivors. So it's sort of assumed that they've already gone through these really intense, stressful events, so they're probably more resilient. So we can use those to propagate further. We increase coral cover by propagating and outplanting the corals. This is through coral gardening, which I think Coral Restoration gave a presentation on earlier. And that's through asexual reproduction, like a plant. You can take little snips from the plant and propagate the plant more, but they're identical genetically. So really important, what we're trying to do is close the loop on restoration by incorporating sexual reproduction and sexual propagation. And the reason why this is so important, because this is where genetic diversity comes from. Genetic diversity is crucial to the adaptive potential of anything, really. If everything is the same, if everyone had blue eyes and a disease came in and infected people with blue eyes, everyone would die. So it's the same with any other species. You need diversity. You need to have individuals that have different traits that might make them better adapted to stress or change. And the best way to get that is through sexual reproduction. Just like with humans, your offspring, your children have different sets of traits from the parents, and every individual is genetically distinct. So we can identify these resilient strains either by going out in nature and finding those resilient corals. We can also use experiments. We can let nature tell us which ones are perhaps the most resilient by putting out different strains and seeing which ones survive during the summer when we have bleaching events. We can also do genetic analyses. So we take samples from the corals and see how related they are. And we actually, in this sense, we don't want relatedness. We don't want to continue to propagate individuals that are so closely related, because we want that diversity. So we can increase the coral cover by propagating and outplanting corals. So this is the coral gardening tenet. One of our main techniques is to use this novel growth method, which is called microfragmentation, and it allows us to produce very large numbers of corals faster. So we take a coral and we cut it into small pieces that are usually similar in size. We use marine superglue, we glue them onto the substrate, and then we can propagate them in our land nursery. So we have a very large land nursery. We have about 100 raceways, that's those tanks there. And currently we're housing about 30,000 coral fragments. And we use this land nursery for propagating and growing a number of native corals on the tree, and once they get large enough, you can outplant them. So our offshore nurseries cover about 1.5 acres of seafloor, and we have about 17,000 coral fragments in them right now. And our coral restoration program, just in the year 2019, outplanted 30,000 corals. And then lastly, we increase genetic diversity and adaptive potential by including sexual propagation and managed breeding. And this is really the focus of my research. So first, how do corals sexually reproduce? Well, they've actually evolved a multitude of strategies for reproducing both asexually and sexually, but most coral species are hermaphroditic broadcast spawners. What does that mean? Okay, a hermaphrodite means you're male and female. So every colony is male and female, produces egg and sperm, and can, in theory, mate with any other colony that's not genetically identical. Broadcast spawning means that they, usually after the full moon in August, they will eject their sperm and egg into the water column, where they will meet and fuse and form embryos and larvae, and then those larvae will settle back down. So there's external fertilization and development. And what's important about this is most of these species only spawn once a year. That's not a lot of opportunities to create the next generation that we need. So why is sexual reproduction so important for corals? Well, it generates offspring that can replenish depleted adult populations. It creates larvae that can disperse to new habitats and establish new reefs. That also serves as a mechanism of gene flow. Because corals can't move themselves, larvae can actually move between the populations and move beneficial traits or alleles between those populations. And again, sexual reproduction generates genetic diversity, which is important for adaptive potential. And it can also support population recovery post-disturbance. So say a hurricane comes through and wipes out a lot of corals. Well, either we have to go out there and plant those corals back, or through sexual reproduction, you'd hope to get the next generation of these self-sustaining corals. So why do we need to intervene, though? Like, if coral reproduction is happening in nature, why do we need to step in? Because of the rapid and widespread loss of coral cover, our population sizes are declining. And as a result, they're becoming small, patchy, and disconnected. And if you're thinking about these corals that emit their gametes into the water column and need to meet, if they're further and further apart, the chances of sperm and egg from colonies very, very distantly located finding each other and actually fertilizing to produce a larvae is very minimal. Also, with the loss of individuals, you get the loss of diversity. And I've already expounded on why diversity is so important. And with the loss of diversity, you also have chances of inbreeding are higher, which can lead to developmental disorders. And then finally, for some species and regions, the sexual cycles are just failing altogether in the wild. There's evidence of chemical pollution, which is leading to developmental toxicity, where the corals are growing fine, but because of the chemical pollution, they are not undergoing gametogenesis, meaning they're not sexually developing. So they'll grow, but they're not going to sexually reproduce. Also, these corals that do the broadcast spawning, they rely on a very specific set of environmental cues to trigger the mass release of spawning so that everything spawns at the same time. And while the environment is degrading, those cues are breaking down. So things are now spawning at different times. They're spawning on different times and spawning on different days. And the reason why this is such a problem is these gametes, these egg and sperm, are only viable for a few hours after release. So if they don't find something, then that's it. And if you have things spawning on different times and days, fertilization is not going to happen. And then finally, in some regions, there's just no natural recruitment. So after the larvae are produced, they look for a place on the reef to settle, and that's just not happening. So maybe corals are spawning, but the larvae are not recruiting back into the population. And one reason, as I said earlier, could be like the macroalgae surface just covering the reef, and there's no free habitat for the larvae to settle down. So as a result, we're seeing very rapid loss and extinction of populations and species. Okay, so what does coral sexual propagation look like? If we're going to help this process, what does that process actually look like? And I have a video. So this last year, I carried out Moat's first round of the full sexual cycle of corals, and this is just a video showing you what that looks like. I should say we had to celebrate because these corals, we have an idea of when they spawn. It's usually a window of a few days after the full moon, but we don't know exactly when. So we have to monitor them every night, and sometimes you're there 8, 10, 12 nights in a row, 4 or 5, until 4 or 5 o'clock in the morning, because they spawn after sunset. So when it does happen, it's really exciting, and when we're actually able to get sexual recruits, it's even more exciting. And what even makes it even more exciting is the fact that sexual propagation for coral reef restoration remains largely experimental. So a lot of the stuff is not figured out completely and has to be independently developed for every species. So a lot of time and effort goes into developing these strategies and interventions. And because it only happens one time a year, it makes it really rare and exciting and important. So, go back to my talk. So then, if we can figure out how to propagate them sexually, what does selective or managed breeding mean, and how can we incorporate that into our intervention? So selective breeding is actually a practice that humans have been using for centuries to domesticate plants and animals, and only recently has it been considered as an intervention for conservation and restoration. But what does it mean? Essentially, it's crossing corals with different traits, usually beneficial traits, to produce offspring with those traits. So this is important to clarify, though. The fact that corals with different traits already breed and crossbreed to produce variable offspring, that already happens in nature. Oh, sorry, I should put it on the, It already does or should happen in nature. But it's not, and I already kind of explained why it's not happening. The sexual cycles are breaking down. So we're stepping in to bring this process into the lab and do it under controlled conditions using restoration genetic principles so that we don't create monocultures or select a few of the best individuals, because I've already talked about we need genetic diversity. So the idea of what we want to do is be able to cross corals from the same species that have different traits. So we've already identified different strains that are disease resilient, that have high bleaching tolerance or thermal tolerance, and could also be fast growing. So what we're going to do is we're going to cross these different corals with these different traits to produce offspring that are robust, resilient, and genetically diverse, and use those for increasing the adaptive potential of our restored populations. So again, we don't want to select just a few of the best individuals. So how can Aquaria help out in our research? Well, there's a lot of money and effort and time has been put into the aquarium trade. Scientists have actually figured out how to use aquarium trade equipment to simulate all of those very specific environmental cues to induce spawning. So now there are a few labs in the world that can actually induce spawning whenever they want. And it doesn't mean that you can induce it more times in the year, but you can probably have one species in 1 month and then spread it across the year. So this really allows us to have our research go forward a lot faster and progress a lot quickly. We don't have to wait that one August for that 1 month in the year for everything to spawn and then try to go out and monitor some species over here, because we have a lot of species spawn at the same time, and it becomes very difficult to try to monitor different species at the same time. So if we can induce spawning when we want in the lab, we can do it during the day. We can convince them day is night and night is day, so that way we don't have to work at 4 o'clock in the morning. And we can also spread the species out across the year, so we can really maximize success. How can aquarium hobbyists play a role in coral reef conservation? I think Frank did a really, really nice job going over sustainable practices and what you should be considering if you're getting into this, whether it's sustainable trade practices, appropriate sources, considering the socioeconomic and environmental impact your collections have, and also become engaged in the coral conservation issue, become aware. And also, I believe Roxanne mentioned earlier that aquarium can serve as sources for gene banking and also opportunities for captive spawning. And then, of course, how can you help? I'm sure everyone here has heard of all the different ways that you can, different strategies you can do to decrease your carbon footprint or be more sustainable or environmentally conscious. But I always say to people, don't try to change your entire lifestyle. That's not reasonable, it's not realistic. Find a few things that you know that you can do in your everyday life just to start with changing. And so in my mind, it also comes down to like behavior modification, right? So you've already conditioned your brain to remember your keys, your sunglasses, your wallet, your purse, your phone when you walk out the door. All right, just throw in your tote bag, just like add that into the list of things that you need to remember to take with you. And try to make small steps towards a more sustainable practice. And then of course you can visit us on Summerland Key, so the Moats Elizabeth Moore International Center for Coral Reef Research and Restoration. We give free public tours on Tuesdays. You can also call to schedule a tour, and you can see our entire operation. You can see our land nursery, you can see our coral babies, you can see all the different species that we're propagating. You can also see how we do the microfragmentation process. And then I just want to end with some awesome photos that Cody Engelsma, our coral reproduction technician, has done using the blue light technology on a number of the different species that we work with, including the sexual recruits. And then I would like to thank you for your attention. If you have any questions.
Big round of applause for Dr. Cook. All right, raise the hands. Who's got a question? All right.
Hi, so you had mentioned the stony coral tissue loss disease and the impact that's having on the reef right now. How is that impacting the outplanting and considerations for species that you're going to outplant from the nursery?
That's a very good question. In fact, there was, after the stony coral tissue loss disease outbreak, there was a moratorium on outplanting because we didn't know if what we were putting out was adding fuel to the fire, if we were just putting out more tissue biomass for the disease to infect and spread. So for a while, we put a halt on things. We did find out that the croperids, so the staghorn and the elkhorn, they are not susceptible to this disease, and they are also not carriers. So we carried on with outplanting, but just redirected our focus to the species that we knew were not susceptible or could cause, exacerbate the problem. And then we also started to put a little bit more direction and research effort into doing this resilience-based restoration. So taking all of our different strains and exposing them to the disease to figure out which of our strains were susceptible and which were resistant. And so we make sure that we only put out the strains that are resilient or resistant.
Anyone else?
I have a question. You probably know some domestic plants, tomatoes, staghorn ferns, whatever, have been exposed to ionizing radiation and other teratogens and even devices to interrupt anaphase 2 in order to end up with multiple sets of chromosomes. Have you heard of any of that sort of work to induce genetic variation in stony corals?
So I think you're referring to, you can use UV to, it's called mutagenesis, essentially. Through mutations, you can get diversity as well. And for those who aren't familiar with genetics and mutations occur naturally during DNA replication, and there are mistakes, and they are the raw source materials for evolution. Yes, there are more extreme interventions and ideas that people have been considering, including genetic modification. So you have a spectrum of interventions. You have some people who think you should do nothing, and then you have some people who think we should be genetically engineering our corals or our strains. And there's a different level of considerations you need to think about when you're thinking about any of those interventions. Those more extreme interventions, I don't know of any that are actually employing them. I mean, I know there is ideas and conversations and possible research going into it, but that's something at Moat that we are not engaged in. And we make sure that everything we're doing is as natural as possible. So what we're doing by crossing corals, that already happens in nature. Sexual reproduction already happens. It's just not happening efficiently or effectively. So we are helping the process by bringing it to the lab. But yes, that is something that if we get to the point where all of our corals are gone or all of our coral reefs are gone, maybe all we have left are to think about artificial reefs or really controversial extreme interventions like genetic modification. I hope we don't get to that point, but everything has been considered.
Are you aware of how far along we are? Do we have full mappings of various popular strains, full DNA mappings? And
like genomes?
Yeah.
We have a number, but there are thousands of species. I would say the most, within the main reef building species, the genomes are mapped.
Okay. Anybody else? No?
So Martin Moe and his wife Barbara, they pioneered some of the aquaculture techniques for bringing back the diadema and alarm sea urchins to provide habitat for planula for the settling stages of stony corals. Do you folks have any actual tie-in with that?
So I do know that part of the problem with the macroalgae covering the reefs is the die-off of the diadema, and I do know there have been, I think a few of the people who've tried to cultivate and culture the diadema. I think FLAC is actually trying to cultivate them right now, but it's been harder to actually produce them in large numbers to have any sort of effect for putting out on the reef. I do know we do collaborate with some other scientists who are trying to find a cheaper, easier, better alternative than diadema, which are large king crabs. And so they are faster growing and easier to cultivate in large numbers. And so there are studies going out right now to look at the effect of their presence on cleaning the reef and how that might contribute back to like a positive feedback loop with coral recruitment. So there are people out there trying to investigate different ways for, controlling the macroalgae problem on our reefs.
Someone else?
Hi. Do you maybe foresee this being something in the future to where aquarists all over the world would be able to help in promoting like the reproduction in their own aquariums, and maybe being able to set it up where they have some, plugs or something in the tank that the new corals could start to develop and maybe be able to send in to places like yours that could then help repopulate the reef?
That's a great question, and that's something definitely that has been considered. Even given this stony coral tissue loss disease outbreak, afterwards there was a movement, the rescue coral movement, where we went out, or not we, but a large consortium of people went out and rescued a bunch of corals to make sure that we didn't lose species entirely. And some went to holding facilities, labs and whatnot, but some of them also went to, I believe, private holding facilities. So it is very realistic possibility that in the future, as things continue to decline and degrade, that we might need to rely on aquarium hobbyists or people who have their own tanks and their own corals. There would definitely have to be an understanding of what the history of those corals went through to make sure that, again, we are following proper restoration genetic principles and there's not necessarily inbreeding and what's going on. But that is a great question and a great idea, and that's something that I do see as the aquarium trade having a potential place in the future with coral reef conservation by doing captive spawning. Get with someone who can, whether that's a person or a scientist or a facility that might be able to give you some ideas of what you should do if your corals spawn. Have some substrates, have some plugs, have that connection with a facility that you can communicate with. So in the future, if we need to draw on the genetic diversity that the aquarium trade has, we already have that in place.
Great question. Anybody else?
So is most of the coral spawning, once you have the fertilized eggs, is it more right now just for research to see like which mixes are doing well, and is it like going to be on a larger scale where it just goes out and once it's like established, is the end game to try to get a ton of stuff out there and just see what happens? Or
That's a great question. Our end game is to get and actually this should be the end game for any coral restoration initiative. The end game should be to get our restored populations to a sexually mature, self-sustaining state as soon as possible. Because we can continue to outplant corals all day long, but we need these populations to be self-sustaining and doing it themselves. So right now, that's a great question. Right now, because it's not these sexual cycles are not happening in the wild, we are developing the protocol, then we want to upscale. So we won't necessarily do, right now we're doing individual crosses to see which ones are compatible, because for whatever reason, some strains are not compatible and you get failed fertilization. And we have a number of different strains, which we call genotypes. And right now, that's what I'm doing in the lab. I'm sort of testing which ones work and which ones don't. But that's one strategy of crossing two parents. You can also just do a batch cross where you take all of the spawn from all the corals that spawn, put them together, let them fertilize, and then seed those back onto the reef. But eventually, we want the restored populations to become sexually mature themselves and spawn themselves. The other problem, though, is that some of these strains that we're finding through my research is that they spawn at different times and different days. And something I found really interesting this summer was that our disease-resistant strains did not spawn after the August full moon when they were supposed to. They spawned sometime before the September full moon. And this is a problem because if we're outplanting varieties next to each other that are not going to spawn at the same time, that's going to, again, lead to failed fertilization and population recovery. So we actually need to know when these different strains are going to spawn so we can effectively evaluate our outplanting design and make sure that we outplant cohorts next to each other that are more likely to spawn at the same time.
Anyone else? Sorry?
Is there time? I have a question. Whatever happened to the cross between Palmyra and cervicornis?
Prolifera?
Yeah.
So I have not seen Prolifera on the Florida Reef Tract. It's in the Caribbean. And so, I'm sorry, what's your name? Bob. Bob was asking about the hybridization between Acropora palmata and Acropora cervicornis, which is the staghorn and the elkhorn. They can hybridize to produce Prolifera, which is actually a really important process because hybrids can actually sometimes be fitter or have like adaptive beneficial traits that the parents don't have, and they also can colonize habitats that the parents can't have. So in this case, hybridization can be really important. I have not seen Prolifera, but that's actually something that I would like to be able to do in the future as we develop our managed breeding program, is to hybridize Prolifera. And what's cool about that is that you can dictate the morphology of the offspring by who the parent is. And so if the mom is elkhorn, it'll look more like elkhorn. And that's important because elkhorn is actually more resistant to hurricanes and physical forces. So we can actually generate offspring that have the genetic diversity, they're hybrids, and we can sort of manipulate their morphology to make sure that it's a more structural sound colony.
Anyone else? I have a quick one for you. Has it ever been observed in the wild, where either collecting or through your process, corals becoming sterile?
That is a great question. And yes, that's also something that I discovered this summer. We have large restored populations of staghorn. In the last few years, I've been going out and doing large-scale sexual maturity assessments. So I go around to the population and I crack the coral and I sample and I look inside for developing gametes right before the August full moon. And we've shown that some of our outplants become sexually mature within 2 years. But this outplant population is now 3 years old, and I only found 2 out of 92 corals that were sexually mature. So this was a very surprising observation result. It also made me very worried. So I asked then, what's going on here in this environment? And so I'm actually just started a reciprocal transplant study where I took corals from our affected site where they're not sexually mature, and I swapped them between our spawning nursery, which are sexually mature, and I'm going to monitor their growth or survival and sexual maturity over time, because one hypothesis is that these corals, even though they're large, they're twice the minimum size needed to become sexually mature. So usually about 30 centimeters is the size when a colony will become sexually mature, and these are like 50, 60 centimeters, and they're not. So one thought is that there's chemical pollution in the water, and there have been something called zombie corals that people have identified, where these corals they're large, they look healthy, they don't have disease, they're growing asexually, but they're reproductively sterile. Essentially, they are not undergoing gametogenesis and reproducing sexually or developing sexually. And one hypothesis is that it's connected to chemical pollution. So it's developmental toxicity. So what I've done is I've taken water and sediment samples from those 2 different sites. They'll be subjected to analytical chemistry to see if there is evidence of chemical pollutants in the water.
Very interesting. Thank you. Anyone else? Once, twice? Big round of applause for Dr. Cook. And again, a big round of applause for Waterbox, because we have had such a unique, interesting group of people coming up and talking throughout the day. These are really topics that you're not getting every day. You guys are asking great questions, educated questions, and it's been really kind of cool and unique to see such a broad spectrum of topics, and you guys sitting here through the day absorbing this and do it. So again, a huge round of applause to Waterbox for putting together such a diverse group of speakers and really bringing an important day together. So you guys again, oh no, you don't, you got about 10 minutes. You can take a quick little break, and then we got the closers coming up. So we got the heavy hitters coming in to close this